All right, so question number one, what are two of the three tenets of the cell theory? And first one is what? And I guess it doesn't need to be any particular order.
All cells come from pre-existing cells.
Okay. All cells come from pre-existing cells. So cells arise from other cells. Cells undergo cell division and give rise to new cells. Okay, what else?
Cells are the building blocks of all things animals.
Okay, they are the building blocks of all plants and animals. Now, I'm not sure why your book doesn't include things like fungi, bacteria, protozoans, because they're made of cells as well. But, whatever. And the third one. Something we kind of addressed when we looked at the organization of living things back in chapter 1. Okay, so we said that cells are the smallest level of organization that can be considered living, and that is because they have the capability of performing physiological functions that allow that cell to maintain homeostasis, essentially. So, cells, smallest level of physiology, kind of a shorthand there. So because cells are able to physiologically respond to their environment and change and all that stuff, that's what that means. And that also relates to why cells are the smallest living thing. Okay? Right, any questions about cell theory? I'll take questions from the notes when I get through this. We'll go back, and if you have any specific questions from the notes, I'll answer those then. All right? Okay, question 2 then. Was about the basic structure of cells. And this little concept map here basically shows us that the cell can be broken down into a couple different parts. And that would include the plasma membrane and the cytoplasm. But the cytoplasm is kind of a larger concept, and we can break that down into the cytosol. So how is the cytosol different from cytoplasm?
It's the liquid portion.
Cytosol is the liquid portion, which includes, well, cytoplasm includes the liquid portion of the cytosol, organelles, ribosomes, mitochondria, endoplasmic reticulum, lysosomes, peroxisomes, and inclusions. Any idea what inclusions are? Inclusions are any of the molecules and chemicals and things that are produced by the cell or brought into the cell that are found in that cytosol. Now, one of the things that's not listed on here, but is still an important part of the cell, which we could kind of include under cytoplasm, and honestly, I don't think it gets as much respect as it should in an intro to A&P class. That's called the cytoskeleton. And basically what that is a variety of different proteins of different sizes that allow the cell to maintain its shape, give it some stability, also allows the cell to change its shape when it needs to. Allows the cell to attach to other cells in certain situations. Allows substances to actually be transported like a railway along these specialized proteins from one place to another in the cell. And if we have time, I'll try to show you a little video today that shows you how that cell works, the inner workings of a cell. All right. Any questions on number 2? Okay, number 3. List 2 of the organelles found in a cell. Well, keep in mind that not every single cell in the body has every single organelle. So what we have is what we call the composite cell. This is what's shown in your book. It's a generalized cell that has everything in it. But not all cells have every organelle in it. Some cells don't have nuclei. Some cells don't have mitochondria. But basically, you could have listed the mitochondria, lysosomes, the Golgi complex, the Golgi apparatus, the Golgi bodies. It's all interchangeable. Ribosomes, centrioles or centrosomes. What else did we have on that list? Microtubules, microfilaments, rough ER, smooth ER. I said don't include the nucleus, okay, but that technically is considered an organelle as well. Now your book did go through and look at some specific details. It talked about the mitochondria, it talked about the Golgi, and it talked about the ribosomes and the endoplasmic reticulum in a little bit of detail. And we can go through that a little later. All right. Question 4. Actually, let me go back to that. Question 4. Cells with microvilli are likely to be involved in what process or processes? So what's microvilli? Or what are microvilli? Like we see here on this picture, they are extensions of the plasma membrane. So these little brush-like, comb-like structures. Notice they are not separate structures. The plasma membrane actually infolds. Now what does that do to the cell or for the cell? It increases the surface area, which means that there's more space available for what? Absorption or secretion, perhaps. A lot of cells, their job is to produce substances that are exported. And so the more space you have for export, the quicker or more quickly you can get those substances into the extracellular fluid so they can be picked up by the blood and transported to the body. So microvilli are very important in cells that play a big role in absorption and secretion. Please keep in mind that these are not separate structures. They are part of that plasma membrane. And in fact, what happens is the cytoskeleton is arranged in such a way to make sure that those microvilli actually stay that way. All right. Question 5. Function of the ribosomes basically is what? Protein synthesis. It takes the amino acids that proteins are made from and it strings them together. It's what facilitates, it kind of acts as an enzyme and it facilitates the dehydration synthesis reactions that forms the peptide bonds between amino acids. Now, if you didn't understand a word I just said, you need to go back to chapter 2 and review that protein stuff. Because amino acids, remember, are joined by peptide bonds, and that's what strings those amino acids together. Now, on Wednesday, we're going to talk about protein synthesis and look at the process and how that works. All right, question 6. We know that the plasma membrane is essentially a boundary. It's really what defines the cell and keeps everything in. So what is the major organic molecule that we find in that plasma membrane? Specific organic molecule. And I addressed this a little bit back in chapter 2. They're a very unique structure, very similar to a triglyceride, but a little different. Phospholipids. And so what we see here in this picture is we see kind of a close-up of the phospholipids. So each one of these is a phospholipid. And notice that the phospholipids form 2 layers with those fatty acid tails pointing together in the middle here and the phosphate heads out here. Now what we see is that this is the extracellular fluid out here, which is water-based. And inside the cell, this is our intracellular fluid, which is also water-based. Why did this membrane arrange itself in this way? I think I talked about this briefly back in chapter 2. Why is this membrane arranged in 2 layers, this bilayer, with the phospholipid heads pointing towards the water and the fatty acid tails in the middle. What's hydrophobic? The fatty acid tails are hydrophobic. They're water-fearing. But what about these phosphate heads? They're hydrophilic. They're water-loving. So these molecules are going to want to arrange themselves so that the hydrophobic portions are far away from the water as possible, and the hydrophilic are close to the water as possible. And as a result, we get this bilayer, this phospholipid bilayer. Now this phospholipid bilayer is very fluid. It's not this stiff membrane. It's very fluid. In fact, we have proteins that are embedded in there. What do we call the different types of proteins embedded within the plasma membrane? A couple different names. Integral proteins, but we also have proteins that can be classified depending on where they are in that cell. So proteins that are embedded in the membrane, and you can see them from both inside and outside the plasma membranes, are called transmembranal because they traverse the entire membrane. But sometimes we have proteins that are just on the surface, towards the extracellular fluid, or just on the inner surface, inside the cell. Those are called peripheral proteins. Now, the majority of the molecules are phospholipids, but again, we have proteins. What are these little things right here? Specific, very unique, four-ringed structure. Oh, rats, I forgot to take my Lipitor last night. My heart's going to kill me. Cholesterol. I actually did take it. But, cholesterol. Cholesterol is important because it actually helps to stabilize this plasma membrane to make it so it's not so fluid. It, in a way, kind of helps to link some of these phospholipids together so that the membrane, it's constantly flowing and moving. It's essentially almost like a living thing. And so this cholesterol is very important for maintaining membrane stability. Notice we have carbohydrate chains associated with phospholipids and with certain proteins. What do these carbohydrates make up when you look at the plasma membrane? They stick up into the extracellular fluid. These sugars, sugar molecules, attached to proteins and attached to lipids, phospholipids. There's a special group of structures, part of the plasma membrane, that's important for your body to recognize your cells as belonging to you. They're like cell identity markers. The glycocalyx or glycocalyx, however you want to say it. Okay? The glycocalyx is this series of proteins and phospholipids and glycolipids that identify your cells as belonging to you. And that's why we see those. So they're identity markers. So those are important. Notice here, down here, this is the cytoskeleton, or part of it anyway. We have these specialized proteins, these long protein chains that act like ropes or wires that play a role in providing some of that stability for the cell as a whole. So that's what that is. All right, so here's kind of another close-up of some of these proteins that are in there. Some proteins are going to have a hydrophilic region. Some proteins are going to have a hydrophobic region. You notice on these proteins, what level of structure would this spiral be on this particular protein? And we have 4 levels of structure in a protein. We have level 1, which is the order of the amino acids. We have level 2, which is the alpha helices or the beta sheets, the way that the chain folds or bends in certain ways. And then the entire shape of the protein is the 3rd level. So we see some alpha helices here. Now, as I said back in chapter 2, I mentioned briefly that proteins by far have the biggest diversity of roles that they can play in the body. These are just some of the roles that these proteins can play in the cell itself. And I think your book goes into a little bit of detail about these. Some of these proteins act as adhesion molecules that allow cells to stay attached to others. Talked about that glycocalyx and cells acting as identity markers. What's a receptor? Receptor is a structure, more than likely a protein, that is going to receive some type of chemical messenger, that when that chemical messenger attaches to that receptor, to that special spot on that protein, that active site, it is going to change the behavior of the cell. We also have enzymes that can be embedded within the plasma membrane as well. Okay, question 7. Processes called the citric acid cycle and the electron transport system take place in what organelle? Kind of looks like this. Has a double membrane. We have an outer membrane and an inner membrane. The mitochondria. And what's kind of the other name or nickname for the mitochondria? The powerhouse of the cell. Why?
It provides the ATP.
It's the mitochondria that makes the majority of the ATP needed by that cell. And the citric acid cycle is one way that we break down the glucose molecule to get energy that can then be used to make ATP. Now, why would this inner membrane have this particular shape characteristic? What is this doing for the membrane, the inner membrane, by having this? Increases the surface area. And the reactions that allow ATP to be made in large quantities actually take place across this inner membrane. So if we have more membrane, then there's more places for these reactions to occur, and theoretically then we can make a lot more ATP per mitochondria. So in cells that really have a lot of energy needs, we're going to see more mitochondria in those cells. In cells that have very low energy needs, like red blood cells, they don't have mitochondria. They actually rely on a different process to generate the ATP that the cell needs. So again, not every cell has every single organelle. Okay.